Let's go back to hearing regeneration, tissue engineering. You're right that if you're a reptile or a fish, chicken, there is a gene that gets switched on that regrows the hair cells. So those species will regenerate hair cells. They will regenerate hearing. That gene is switched off in humans. And so there's a lot of work trying to switch it on.
Yeah, just
and you can imagine.
flip the switch,
right.
man.
Can you please switch it on? Everything will be good now. It doesn't work that way.
Hey guys, welcome back to the Dr. Cliff Show. I'm Cliff Olson, Doctor of Audiology and founder of Applied Hearing Solutions in Phoenix, Arizona. And I'm joined today by a very special guest on the show, Barry Seifer, co-founder and CEO of SoundBites. Barry, thank you so much for coming all the way out here to Phoenix to talk with me.
It is my pleasure. Thank you for having me.
So a lot of individuals who watch the show here, they're probably thinking, well, you know, Cliff, you get emails from everybody all the time, probably asking to come out on the show and all of that, and that is not how this relationship started. I actually saw some information about you guys online, and I reached out to you, and we had a really nice conversation about, what, 2 months ago?
It was quite a while ago, and it was a very unusually good conversation. We as well get contacted by other folks. The conversation I had with you was different, and it's frankly the reason I'm here.
Awesome. Well, again, thank you so much for coming out. So for everyone who's watching right now, we'll get into the history and all of that of SoundBites, but can you just kind of give us an overview of what we're actually talking about here today in terms of the product that you guys have?
ACEMg is provitamin A, which is beta carotene, and vitamin C, and vitamin E, and magnesium. So you put those four letters together and you get ACEMg.
Excellent. And so the intent of that, do you guys call it a nutraceutical?
Interesting. We call it hearing preservation.
Okay.
Other people either call it a nutraceutical or a supplement.
Okay.
Or we call it a biomedicine because that's really what it is. It's a biomedicine comprised of antioxidants and a vasodilator, which most people don't want to say. So the product is a nutritional that was developed using the translational research process that you would use if you invented a new drug called a unique molecular entity. And we used that process because the formula, ACEMg, was developed in a medical school. And so we progressed the science out of the lab using translational research to prove what we proved in the lab, which was that we could preserve the hearing of a guinea pig. And we wanted to prove that it worked as well in a human. And that took 15 years.
Well, I would say that guinea pigs need to hear too, so that's very important. But you know, this term of translational research is basically taking laboratory findings and then translating them to actual humans, right?
Right. And most people understand translational research by the phase numbers, phase 1, phase 2, phase 3. And what you get at the end of that is an FDA approval and a drug code. And what happens after that is that the prescription is then able to be given to you and monitored by a physician. And if you are fortunate to have health insurance, your insurance pays part or all of that prescription medicine. That's what translational research is. Now, what's important for us is that we went through that process and we're still going through it, but a nutritional does not have to do that. We did.
And I have to ask, so like, where did you guys even think of combining these different, nutrients together?
The answer to that question is 20 years long. So bear with me. It didn't come out of mixing things in a bathtub one evening and saying, hey, let's try this. No, it was not like that at all. So the original motivation was not mine. It was my late co-founder, Joe Miller, who became the director of the Kresge Hearing Research Institute at the University of Michigan Medical School in the late 80s. And he was a biochemist by training. And at that time, the conventional wisdom about hearing loss was that it was a mechanical failure of the physical structure of the ear. His background was in biochemistry, so he started to look at what was happening inside the cochlea, which is the part of the ear, the hearing organ that changes hearing waves into signals that go into the brain. He did that work for about 18 years and discovered that the root of inner ear hearing loss, which is called sensorineural hearing loss, the damage to those sensory nerves, is increased oxidative stress that is triggered by a high rate of metabolic energy that is necessary to process sound, especially sound that is loud. So the original motivation was noise-induced hearing loss. How can I stop or mitigate noise-induced hearing loss? By 2003, he figured out what the root cause was, and that's when they started looking at antioxidants. Now, the Israeli military had tried high doses of magnesium with their fighter pilots. That seemed to work. Other people had been looking at antioxidants. They tried all sorts of antioxidants. And to finally answer your question, they put 3 antioxidants together with magnesium and got what is called an unexpected synergistic effect. And I can explain why. When you have an excess of metabolic energy in those hair cells, which we'll get to in a minute, I imagine, and you get a vast increase in the production of reactive oxygen species, the cells have built-in antioxidant systems that neutralize those toxic molecules. But when you overexcite them, they generate so much extra antioxidants that they overwhelm the onboard endogenous antioxidant systems. Those are the antioxidants that the body makes. So then the cells start looking for exogenous or antioxidants that are in the food. And so the idea was, well, feed them. Give them all the antioxidants they need. And in addition, what happens to those cells is that the reactive oxygen species, the ROS, lock down, constrict the very scant amount of blood that is in the inner ear. So they get trapped. So the magnesium is a vasodilator and it keeps the blood flow going. So what they proved when they actually did the lab studies was that they could reduce the noise-induced hearing loss of a guinea pig by about 75% and protect the cells from damage up to 31 decibels. That's huge.
That is absolutely huge. The thing that I found really interesting when I was looking through all of this is that you can, even in this graph that we're looking at, is that you take the 3 vitamins and you don't see the massive reduction, right? And then you just add that magnesium in and then boom, you see this massive effect that is more powerful than just those vitamins alone.
Correct. So that paper documenting the study, the lab study on the guinea pigs, was published in 2007. The research was conducted in 2005, and the paper was peer-reviewed and published in 2007. And the NIH and the University of Michigan said, you know, you really ought to get this into translational research because it's safe, and there's an enormous need for this.
Right.
And that was the trigger for getting this started into translational research.
You know, it's interesting. I served in the military from 2002 to 2006. We briefly talked about that before we turned the cameras on here. And noise-induced hearing loss obviously is huge in the military. I went on several combat tours. You know, there's not always an opportunity to don your hearing protection when you're there. A lot of, you know, my buddies from the military and then other individuals who I went to high school with who went into the military, now they're all reaching out to me saying, hey, you know, I noticed that you're in the world of hearing loss and hearing aids, and it's everybody who I served with and everybody who I know that served is all going through noise-induced, you know, hearing loss at this point, where it's negatively impacting their lives.
It actually brings tears to my eyes. I hear these stories so often, and it is so sad, and it's so ubiquitous, especially in the military. And many noise-exposed professionals face the same issue. And as you said, you go into battle, you go into the field, you go into small arms training, you go into large arms training, right? And the first thing you do is you put on hearing protection. And the first thing you do after you have your hearing protection on, when you have to talk to someone, is you take your hearing protection off.
Yeah, exactly.
So that's a problem.
Yeah.
It's a very big problem.
So now, how did you connect with Dr. Joe Miller? I mean, you guys were friends prior to that.
Correct.
And he probably, you know, over a beer or something like that, kind of shared this information with you. Like, how did that all start?
So it started, you're right, we were friends. And he was a scientist, and I was not. But what I was doing is trying to find a way that I could take what I knew how to do, which was essentially business strategy, and apply it to science to accelerate, in my words at that time, accelerate the translation of science out of the lab into the market. So Joe was my advisor, my scientific advisor. I was not looking at his science and he was not looking at me.
Yeah.
I was out in the country looking around for all these things, and I would come back to Joe and say, so what do you think of this? And he'd say, yeah, maybe, could be. We did that for several years. And then we sat down one day and we said, maybe we ought to do this together. And we made a deal. The deal was we would progress this business together as long as it didn't interfere with our friendship. And if it did, all bets were off. And we became very, very good friends.
And what year roughly was that you guys had that discussion?
It was August of 2009 when he said, how about we do this? And then in June of 2010, we started working together. And then 9 months later, I should stop and tell you that we were both from California.
Okay.
So we're both transplants from California. We both went to Berkeley. And so we ended up in Ann Arbor together. And when we started moving this business together forward into research, we said, why don't we move back to California? And then one of his former postdocs called from Spain and said, Joe, your science is perfect for a European Commission grant. It's called a call for entries or an open call. And take a look at this grant proposal and see if your science fits. And it did. And Joe said, we ought to write a proposal. And I said, oh no, aren't we going to California? You realize it's an 11-hour flight from California to the EU? And but we applied. We went through the first phase and were approved. And then we got approved in the second phase. And I moved to Europe.
Did you both move to Europe, or did he stick back at Ann Arbor, Michigan?
He stayed here. I moved.
Or did he go to California then? And,
no,
and you had to go to Spain?
Joe stayed in Ann Arbor. My wife stayed in Ann Arbor. And I was one of 5 principal investigators on this multi-year, multi-million-dollar, multi-million euro grant that funded us for more clinical research and basic research, as well as giving us money for the commercial strategy, which is very difficult, if not impossible, to get in the United States. So that was very useful. It was not an easy journey to take, but we learned an awful lot, and we credit the European Commission for understanding that the problem that you said earlier is so ubiquitous, affects hundreds of millions of people in the EU, and they very much wanted to protect the hearing, especially as people age. And they saw our science and said, okay.
So I'm super interested in this kind of progression with how everything went with this. First and foremost, I'll say this. When it comes to nutrients, vitamins, we're not talking pharmaceuticals here. Right. A lot of companies that will, produce a supplement and put it out to market, they don't really care to do research on it. They might look and say, oh, these products are safe based on what the FDA has said, and then they'll go and they'll make their claims about the product. You guys chose not to go that route.
Right. That's one of the reasons it took us 36 years to get here. I mean, think of it this way. This conversation took 36 years.
Yeah.
We're in our 37th year. So we're the world's oldest startup, I can imagine. You raise a couple of very good points there. The first is, I'm not sure that people who make supplements don't want to do research. I think that they are hobbled by the cost of the research and the time it takes to prove something. That's point one. The second point is, it's extremely difficult to prove cause and effect when you put an exogenous antioxidant into a human body. The human body is hideously complicated. And so people will say, well, you ought to take this for that or this for that. And I don't have to tell you or anybody else, there are claims everywhere for what you ought to be doing to reduce inflammation, because everyone now knows that inflammation is bad and antioxidants are good. That's conceptually accurate.
It's kind of funny because, my background from undergrad studies is exercise science. And so I look at, in a lot of ways, from a physical health and then physical performance standpoint, as an athlete myself, that there was studies that came out that showed that if you took too many antioxidants in close proximity with, physical, like stressful exercise, it was actually a bad thing.
It is. I'm glad you mentioned that. Yes, there are pretty robust studies, which you can study because now you're just talking about antioxidant intake and metabolic output, right? So you can measure that, and you can measure that at the level of the organism. But you can't measure the impact on the stomach or the spleen or the this or the that, right? The beauty of what Joe did was that he focused on an organ called the organ of Corti. The organ of Corti does one thing. It transduces waves in liquid, in liquid in the cochlea, which is incidentally half the size of the pinky fingernail, and it transduces those into electric signal. That's all it does. And so he was able to do histology. He was able to count cells. He had a lab that was using its scientific investigation tools to figure out what actually happens to these cells. You can't really do this with larger organs. And so he was very fortunate to be able to isolate the work of an antioxidant, the work of vasodilators on the cells of the guinea pigs.
Well, it's super interesting too, because like you think of, athletes trying to get better at physical performance, putting the body through stress, like the muscles through stress,
right,
and then having those muscles get used to that stress and then get stronger because of it.
Right.
That's great for the muscle anatomy and all of that and physiology. But when it comes to the cochlea, I mean, we don't have the ability to have this oxidative stress cause damage to these structures inside of the cochlea and then have that become stronger because of it.
Because we're not talking about a muscle. We're talking about nerves.
Right.
And nerves are different.
Totally different.
And I said earlier we were going to get to the hair cells.
Yeah.
So that's actually what we're talking about here. We're talking about nerve cells. We're talking about unbelievably complicated and elegant cells that have evolved into the human body in 250 million years. Okay, humans didn't evolve overnight either, and so we evolved very slowly, and the hearing system evolved from other species into humans. So what we have as humans are these roughly 15,000 nerve cells in a pair of organs that do this absolutely remarkable work. And the job that I think we have is keeping them healthy. That's what Joe and I did. That's what Joe did. And that's what I promised to do at the end of his life, is to carry that work on, because it's really important.
Yeah, I want to talk about a little bit here before we kind of get into the rest of the history of the company and where we're at today with everything that's going on and all the research that has been done around your guys' product. But, a lot of people, like, the hot topic is like hair cell regeneration. Since we're talking about that, I feel like it makes sense to bring it up now.
You bet.
Hair cell regeneration either through molecular therapies, so literally injecting drugs into the middle ear cavity, have it permeate the round window and make its way into the cochlea and try to stimulate these progenitor cells inside of the cochlea to regrow new hair cell structures. You have the what I think everyone thinks of is stem cell therapy. Like, can we grow stem cells inside of a lab, regrow these hair cells, transplant them into the cochlea, and then somehow get them to innervate and actually create this neural response from this movement of the fluid inside the cochlea.
Nicely done.
Right. If they could do that, that's. I've read a lot of research on the challenges of even just doing the transplantation. Like, you can grow the stem cells all you want to in a lab. You want to translate that into the cochlea or transfer that into the cochlea. That is a totally different ball game, because it's a really harsh environment. Plus, you have to drill a hole into the cochlea to try to get them in there and then get them to innervate.
You can use a very fine needle.
Yeah, I guess so. And then you have this idea of genetic therapy. I was listening to a podcast years ago about this CRISPR drug or virus really that they could program to do something genetically altering and then inject that into you and then have that go in and essentially you take the avian like birds, they can regrow hair cell structures inside of their ears. So if they get hair cell damage inside of their ears, no problem, they regenerate it and then they can hear again. Trying to replicate that type of genetic, a characteristic inside of a human, that's what everybody thinks about here. I think that we should be thinking primarily like the way that you're thinking is preservation. And I know that, a lot of people think that audiologists like myself are like, well, you don't want to prevent hearing loss, you want people to have more hearing loss, so you can treat more people with hearing aids, cochlear implants, auditory brainstem implants. That's what everyone thinks of. I think we should start transitioning the thought process of this to two things. Number one, the preservation of hearing, whether that's through the preservation of the hair cell structures themselves or preventing this oxidative stress that prevents the ear from working as well as it possibly could, and maybe even having a little bit of, dare I say, restoration or regeneration of the function of the cochlea, even if there are missing hair cell structures. Would you agree with those statements?
Well, you've said an awful lot. Let's go back to hearing regeneration, tissue engineering. You're right that if you're a reptile or a fish, chicken, there is a gene that gets switched on that regrows the hair cells. So those species will regenerate hair cells. They will regenerate hearing. That gene is switched off in humans. And so there's a lot of work trying to switch it on.
Yeah, just
and you can imagine.
Flip the switch,
right.
Man.
Can you please switch it on? Everything will be good now. It doesn't work that way. And there is so much wonderful science going on, as you say. But there is a very big gap. And we are the poster child of that gap between proving something in the lab and getting it into a human, knowing that it works. Those therapies are very far away. However, those scientists that are working on that are showing us things that are actually impacting our product as well, things that we didn't know. That's why science is so wonderful. So what is true today gets better as time goes on. Here's the example. For close to 20 years, I thought, Joe thought, and the researchers thought that what we were in business to do was, as you said, preserve the biological function of the outer hair cells, and those cells are the cells that are the first in line. They're on the front line, and what they do is they amplify the signals that are coming into the cochlea, and they separate them into frequencies, and then they pass that information to the inner hair cells, and the inner hair cells transfer that information into electric signals and then pass them through the spiral ganglion nerves into the auditory nerve and into the brain. We knew that ACEMg had its best chance of working on those outer hair cells, and that's what we went out to prove, and we proved it. 75.3% of people who were hearing impaired, who took ACEMg for every day for 2 years, within 6 months, 75%, as I said, either stopped losing their hearing, and half of those people, about 37%, actually regained some hearing function. That's not supposed to happen.
Right.
When the data analyst gave us that report, he said, okay, this is what happened. We completely forgot about the fact that 37% of that 75, or half of that 75%, stopped losing their hearing. And we said, wait a minute, are you saying that people actually gained hearing function? Yeah, we did.
So what's crazy about that, so I have this conversation all the time with patients because I still work in the clinic. I'm treating patients. Ironically, literally yesterday, had a patient come in for their annual visit, which is where we do an updated audiogram for them. And unfortunately, this gentleman's hearing had declined from the previous year that we tested him, required us to make programming adjustments in his hearing aids, so on and so forth.
Yep.
But the question that he asked is like, is there anything that we can do to slow down the progression of my hearing loss? And knowing that you were coming in here today, I'm like, you know what? Yeah, I think there potentially is something that you could do that is very low risk with the potential of high reward of getting some preservation there. And then maybe even having a little bit of restoration of hearing ability. So can you talk to me a little bit about that particular study? Because otoacoustic emissions, the distortion product otoacoustic emissions, is what you guys use to determine this. So that was an objective testing of, or not voluntary, like I'm raising my hand when I hear the beeps, but that particular study that you're referencing that showed the not just preservation, but improvement.
Right. So what we say to people like your patient is, now I can tell you, because of that study, I can tell you that the odds are 3 to 1 that we can help you. I couldn't say that last year. Now I can say that. I can say that to the world because we have the data. And your point is, we got the data using a very different way of measuring hearing than is typically done in an audiological practice. The pure tone audiogram evolved out of and was standardized out of his need to find ways that soldiers returning from World War II could be fitted with hearing aids because they were all so severely hearing impaired, and there was a crush of demand for hearing aids. So that is still the way that hearing aids are programmed. That way of testing hearing doesn't get to the biological function of the cells that are being impacted when someone is asked to tell you whether they can or cannot hear the beep. So the otoacoustic emissions test is typically done on newborns and infants who cannot tell you whether or not they're hearing. It's a completely passive test. And what it does, and it's brilliant technology, what it does is to put a sound at a frequency into the eardrum and stimulate certain frequencies in the cochlea and then follow it with another frequency that causes a distortion, and then measure what happens when it comes back. And it's brilliant. So we used that technique with people who had a history of having that test every year, which is very unusual for adults. Typically, the conventional wisdom is, well, once you've lost your hearing, it's not going to come back. So why are we bothering to do that test? Well, there was an audiologist in Chicago that had been bothering to do that test, and she realized, and I realized, that was a control group. My goodness, we have all of this data that is objective data that confirms the conventional wisdom that people do lose their hearing over time. Great. Let's see if we can get them to stop. That was the idea.
Right.
And we did.
So interesting here. I know that you can do research in different ways and have different types of controls and all of that. When you guys were looking at that, so obviously you have this massive control group of individuals who had been going through this for 2 years, right? We
had a total, the N number, the number of participants was about 200. So we had 93, if I'm remembering the numbers right, we had 93 in the treatment group, and we had about 97 in the control group. And that control group basically just kept doing their annual OAE test, but didn't get the intervention. And then we asked some of those people if they were willing to take it, and we got those completes. And those people went, they started taking ACEMg as SoundBites, and every 3 months they came back to the clinic, so much more frequently.
Yeah.
And that's when we found that even though we tested these people for 2 years, after 6 months, it either had an effect or it didn't. And once it had an effect, this is really fascinating. Once it had an effect, the effect was durable as long as they kept taking it. But, you know, people are people, and they'd come in and say, oh, are you taking it? Well, no, I forgot, right? And the OAE would show that actually they started to lose their hearing.
They regress again.
They regressed.
Yeah.
And then when they started taking it, it came back.
Interesting.
So it was a wonderful study from the standpoint of getting us statistically valid, objective data on the biological function of those hair cells, the outer hair cells. But here's where it gets really fascinating. It gets to your point about what the hearing regeneration scientists are teaching us. We started reading about this because people who were involved in that 2-year study were coming in saying, Doc, is this supposed to help my tinnitus? And we kept collecting the anecdotal data on that, but it was just too much for us to handle getting statistical data from surveys on patients who were reporting that, but it kept happening. So we started looking at what we could do, and we decided to do what's called a single-arm, non-randomized, real-world evidence study to see if ACEMg worked to mitigate, reduce the severity of tinnitus symptoms and hyperacusis symptoms, which is intolerance to loud noise or normal noise. And so it took us about 6 or 8 months to figure out how to do it. And then it took us about another 6 or 8 months to get it approved and get all the tech assembled. And it started enrolling in December of 2024, excuse me. And the reason we did this was that the scientists were telling each other, that the trigger for tinnitus is not in the outer hair cells. That is the initial trigger, but the impetus, the consequence, is in the inner hair cell to auditory nerve ribbon synapse connection, another set of nerves. And that set of nerves is even more sensitive to damage than the outer hair cells. So, hidden hearing loss or synaptopathy is the new topic. And we didn't realize that until we started doing the research, and now we are actually collecting the data. And so we have this IRB approved clinical trial that you can enroll in. You can talk to me and I enroll you, and if you're 18 years old and you're reporting tinnitus or hyperacusis, you're in. If you don't have any other comorbidities. You can also do the same thing if you just go and buy it. The difference is that, first of all, if you are enrolling in the clinical trial, you have to talk to me, and some people I won't accept because we can't test them. They have certain things that we just can't control.
Right.
But any customer can use the same thing, and we're collecting the data on that too.
So I think a lot of people at this point are pretty interested in this, like, well, how did this product come to fruition? Because I spent a good amount of time watching all the videos that you had produced, which are fantastic. And being someone who does videos myself, I'm like, golly, you're taking a lot of what I would consider to be complex pieces of information and then streamlining it and making it really easy to understand. So credit to you, for doing that. But one of the things that I picked up on was, you guys, you have this combination of ingredients that you want to get into a human, but it's like, how do you get it into the human?
And how do you make an identical placebo, because you're in RCT research.
That's right.
Random control trial research. You're in a drug trial.
Right.
Thanks for asking that question, because that's what took me about 5 years.
Oh, jeez.
And lots and lots of heartache. So for all of your listeners, you are correct. There was a formula. And when you scaled it up from the body mass of a guinea pig with pharmacokinetics, which is the way you do this, you get 1.3 grams of vitamins. First of all, you cannot use that much vitamin A because it's toxic, so you have to use something else. So we first tried chewable mints. That's where the name SoundBites came from, and it stuck. That was a really good bad idea because it was impossible to get everything we needed into one capsule. And we were told that if you're going to do RCT research, you really need one and done per day, because if you have more than that, the compliance in a clinical trial goes way down.
Yeah.
So we had 6. You had to chew 6 mints. That did not work. We had a partner, a pharmaceutical development company that was just wonderful, and they were struggling with us, and they said, well, there's these people over in France that we think we can use to do a liquid, so you could pour this a powder into sachet, and you could mix it up and drink it. That didn't work either. And then we said, how about a liquid that was pre-mixed? And that worked really well. No one could afford it. It only lasted like 12 weeks under refrigeration, and we couldn't use it. And then finally, I was in the Netherlands as the principal investigator, as I said, and I went to the pharmaceutical development arm of a company in Tilburg, Netherlands, and I walked into the room, and the chemist threw a piece of paper across the table and said, "We can do it. It's a soft gel." And I said, "Seriously?" He said, "Yeah, it won't be 1, it'll be 2, and they're sort of big." And I said, "Okay, sign us up." And that's what we did. And it was an identical placebo, which you need for You need to be able to double blind the study. You need to
Right.
be able to say, we don't know what you're getting, and neither the nurses or doctors. It has to be double blind.
Right.
We did
Which
it.
is one of the strongest types of studies that you can do.
That's the gold standard.
Yeah.
That's the gold. Ultimately, we didn't use it. And the reason we didn't use it is that the idea of a randomized controlled study is too expensive to do if you're going to try and find out what we wanted to find out. People lose their hearing over time unless they're exposed to
noise,
traumatic
right?
levels of noise, and then they lose their hearing right
Right.
away. But we didn't have the money. No one has the money to do a 3 or 4 year observational study, double blinded. We had a total throughout 30 years of about $30 million. And if you look at the kind of businesses that are in the drug regeneration business, we're talking about $1 billion or more. No one is going to invest that kind of money because no one dies of hearing loss. They may be disabled, but they don't die. It's not a fatal disease.
Yeah. So let's take a look at the actual, we have a couple of the pills right here. And let's talk about these a little bit here. I think that this one right here, is this the, no, this is the adult one. So we have the adult SoundBites, and you also have a children's SoundBites, right?
Yes.
And the one that I'm holding right here, this right here is the adult one. And the one that looks like a little fish, I'll let you hold that one. The one that looks like a little fish is for kids, but a little bit different in how they're taken, right? So me as an adult, I would take 2 of these soft gel
Correct.
capsules a day.
Correct.
And then, but you know, there's no way a little kid's going to be able to take that down. Right. So we also have this. It's at a lower dosage for a smaller child. And then you actually, twist off or cut off the top there?
Yes, and you'll forgive me if I don't do that.
Yeah, please.
Because
you're on a white couch there.
Inside here, there is what's called a hydrophobic compound. And that's what we finally ended up making. The soft gel is beta carotene, so there is no vitamin A in it. Even though it's called ACEMg, there's no vitamin A in ACEMg. It's beta carotene, which is converted into vitamin A as the body needs it. So in the US and in the EU, there's really no upper limit for it, theoretically. So you can't, it's not toxic. Vitamin A can be. And so we made these because there is a very big problem in the world. We're talking about problems with adults having hearing loss. There's another very, very large problem in the world, and it is genetic hearing loss among kids. And there are, well, in the last couple of decades, there has been a lot of research on genes and gene therapy and genes that cause hearing loss. And what we discovered, and what is actually made right now and can be used by parents with children who have GJB2 mutations for hearing loss, is this. And so when we were in the Netherlands, I knew about this, and I asked the pharmaceutical development company to cheat. I said, can you do me a favor? Can you make me a special batch of little ones for the little ones? And so they had this bronze dye that is used for cosmetics. And L'Oreal actually uses this, and a lot of other cosmetics companies use it. And I don't know if your viewers can see it, but it's got a little fishtail on it. And what you do, if you're a parent, is you twist it, and then you squeeze the content into anything that has fat. It won't mix into water, but it will mix into, say, yogurt. And if you are 2 years old, and you have this genetic mutation that is guaranteed to make you deaf by the time you are a teenager, these work. These work.
So now you can do the same thing with the adult version if you're not able to swallow this
Yes.
for whatever reason. You said you can actually poke a hole in it very carefully and then squeeze that into like yogurt, like you said, something that has some amount of fat in it.
We have a few customers who cannot swallow those soft gels, and so we recommend a seam ripper, not a
seam
pen.
ripper. Yeah,
seam ripper is great.
Yeah, because it actually has a stopping point when you go and you
and
push
it gets
in.
wider. And
Yeah,
so,
yeah, very good.
But beta carotene stains. My close colleague Joop, who you've spoken with in Spain, his son was involved in chemotherapy, and ACEMg works as an antidote to the ototoxicity from cisplatin chemotherapy. And he was in a hurry one day, and he just poked a hole in one of those, and he said, "It stains. I couldn't get it out of anything." Beta carotene stains. Fair warning to anyone who wants to do
Yeah,
that.
don't chew these and have it squirt out your mouth, especially not if you're wearing white pants or sitting on a white couch, right?
Right.
So yeah, this is definitely doable for me to take down each day.
Oh.
And you know, it's really interesting to see how far this has come from, you know, the initial research that was done, going through all the trials, trying to come up with a product, going through all these trials overseas in Europe, then bringing it back here, going through additional trials here, to the point now where not only do you have the adult version of the product, you have the children's version of the product. You can participate in the actual continuing research of this to determine its effectiveness and hopefully get to the point of some level of predictability of knowing who's going to be able to benefit from it, who's not, with a higher level of accuracy. I'm always interested in that concept. You know, we had talked about our modern tinnitus project where we're using tracking biomarkers for individuals with tinnitus. And we eventually want to get to the point where we can be predictive of what treatment is going to be best for certain individuals when it comes to treating their tinnitus. I imagine it'd be really cool to do the same thing with these.
Well, we're doing it. We have this Otis study that I mentioned earlier. It's going on at the, you can go to clinicaltrials.gov, and if you're 18 years of age or older and right now living in the United States with a mailing address, you can ask to enroll. You talk to me. I am the coordinator of the study. You read an informed consent agreement, and you can participate for free for 24 weeks, and everything is done online. You get the product without cost. You take baseline exams. You take a digit and noise test, which we can explain in a minute, which is very different from OAEs. Mm-hmm. You take surveys for tinnitus and hyperacusis and hearing sensitivity. You take the product for 3 months. You go online, you take the same tests again. We send you 3 more months, 12 more weeks of the product without charge. At the end, you take the test again, and the tool in the background does all the calculations, and it shows you what happened.
That's awesome. I'll go ahead and put links in the description of this video.
Please do.
For everything that you're referencing here, including the research and all of that. I'll go ahead and link to that stuff as well in the description here. With that being said, I do want to talk, I mean, there is definitely going to be people who watch this episode here, who look at this and hear us talking about this concept of hearing loss preservation or hearing preservation, mm-hmm. and then maybe even some level of enhancement, restoration, or what have you. And they're like, you guys are full of it. Like there's no way this is snake oil. Like, I mean, there's going to be people out there who are the naysayers. And I mean, how do you respond to that? I know you're doing as much as you possibly can with actual research to prove that this works. But, you know, can you ever truly get through to everybody?
No. And we don't expect to. So it's a really important question, and it's very timely. Trust in science is having a moment, and your viewers know it. And nutritional supplements are part of the problem. And I understand the limitations that companies have when they want to do the research that we have done. We were very fortunate to have backing from the NIH and from the European Commission from 1988 until 2016. The work we've done after that, we have invested in ourselves. Many companies cannot do that. And the US government allows them to sell products that they can claim enhance your health. As long as you are on the health promotion side of your claims, you can sell pretty much anything. You cannot claim what we can claim because you have no basis for it. But that's okay. Many people just want to stay out of the hospital. I get it. I do too. We have struggled, and I think that's a fair word, we have struggled to get the data that puts us in a different category of supplement. And yeah, it did take 20 years to do it. And we're still doing it. Earlier, we talked about phase 1, phase 2, phase 3. Most people don't know about phase 4. We're doing phase 4 studies. A phase 4 study is an open label, post-marketing, single-arm public health study. You can do them with drugs. You can do them with OTC drugs, and you can do them with supplements. And that's what we're doing. That's what the Otis study is. That's that 24-week study we talked about. And the beauty of that is that it takes the responsibility from efficacy to effectiveness. So forgive me while I go down this little rabbit hole with you. If you have a drug, your purpose in getting your drug approved is to show that what you make is better than the current treatment. And you do that statistically, and you do it with RCT trials because you have a blinded control group and a blinded active group, so no one knows what they're getting. And you need to have a statistical difference between those things. At the end of that, you have what's called efficacy. In this group, this was the level of efficaciousness, if that's a word. Then your product gets approved, and physicians prescribe it. That's when you see the effectiveness, because there's no more control. And there's generally a gap between what you proved in an RCT trial and what happens in the real world. We're in the real world. We want to prove how well this works for you and for you and for you. It's going to be different because you're all different. However, you're non-randomized. Anyone can show up because it's safe. And that is a huge benefit. There's nothing wrong with this. There's no adverse event that's going to happen for you. And so we have this phase 4 trial, and we want to run it forever. Right now we're running it in the US. We want to run it internationally. We want to be able to improve the diagnostics that we do. And that is the best way we know of to overcome this inherent skepticism, which is totally understandable. How can you prove this works? How do I know it's going to work for me? Those are great questions, and we can answer them.
You know, you kind of, my last question I usually like to ask on the show here is, well, what's the future look like? And you pretty well just laid it out right
Part
there.
of it.
Right?
Part of it.
A lot more fun stuff coming on. Anything that you can talk about?
Yeah, I can. I can talk about it. So people want to know how well this works, and we want to help them. And this gets into your business. This gets into audiology. What's the future of audiology, right? Our view is the future of audiology is hearing preservation. Right now, audiology is hearing remediation, right? So if you want to protect your hearing, you wear earplugs. You do, but you don't, because you can't wear earplugs all the time. Instead, what you do is you wear noise-canceling earbuds, right? And only recently have those noise-canceling earbuds come with noise limiters. So it's possible to hurt your ears with those in your ears. Mm-hmm. It's also possible to hurt your ears with hearing aids.
Right.
It is. So what's next? Well, what's next is to find a way, and we think we are in the lead here, to protect the normal biological functions when you don't wear hearing protection, and even when you do, and then measure it. So we are in the process of making a device that you can use that is compatible with what we make as the intervention, so that you can monitor and then assist your hearing dynamically over time.
Very cool. Very cool. Well, like I said, I'm going to make sure that I have all of this stuff linked in the description so you guys can do your own research yourself. But Barry, I really appreciate you coming all the way out here to Phoenix to have this conversation. I have a feeling this probably won't be the last time that we talk.
I do too.
Yeah.
This has been wonderful. Thank you.
Excellent.
Thanks for the opportunity. You have been very gracious and quite disciplined in the way you have approached the conversations with us. And I know that your patients are benefiting from it too. Thank
I
you.
appreciate that. And you actually came, you brought me a couple of gifts. You brought me a couple hats, which are fantastic, but every time someone comes out to the Dr. Cliff show, I bring them a gift as well. So this is for you. You may have noticed that I've been drinking out of a nice Dr. Cliff show mug on my table here, and you've been drinking out of that crummy Fiji bottle there. But that's my gift to you for coming out. And again, thank you so much.
Thank you.
Guys, if you want to learn more about what Barry and everyone over at SoundBites is doing, make sure that you go down into the description of this video and check out all of that information. If you guys like this episode, do me a huge favor, click the like button. And if you are not yet subscribed to the Dr. Cliff channel, go ahead and do that as well. And we'll see you in the next video.